This is the first lecture in Part 3 or Unit 3 for microbiology. Metabolism is the chemistry occurring in a living cell. Many microbes have the same or similar pathways as plants and animals. Others will have diverse and very specialized metabolism. This section is broken down into multiple smaller units so you can study the individual pathways separately. There are 8 elementary statements which summarize the overall process of metabolism. First is that every cell acquires nutrients. Cells need energy and building materials, and these are acquired through nutrients. Second, metabolism is required or requires energy to fuel the process, and this is supplied through light energy or breaking down nutrients. Third, the energy derived from these processes is stored in the bonds of ATP. Enzymes are used to control reactions, and the nutrients are broken down into precursor metabolites, or the building blocks. ATP is used along with enzymes to control the reactions to build the products out of the precursor metabolites. ATP is used to link the building blocks through polymerization reactions, and cells grow by assembling these macromolecules into structures. Finally, the cells reproduce when they are about double in size. There are also a few main types of reactions. Catabolic reactions break larger products into smaller molecules and release energy as ATP. Anabolic reactions synthesize larger products from the smaller molecules and require energy from ATP to fuel the process. Redox reactions, or reduction-oxidation reactions, involve the transfer of electrons. Electrons will move from a donor, which is said to be oxidized, to an acceptor, which is then said to be reduced. There are a few key electron carrier molecules used in the synthesis of ATP. These are NAD+, or nicotinamide adenine dinucleotide, NADP+, or nicotinamide adenine dinucleotide phosphate, and FAD, or flavin adenine dinucleotide. When ATP is produced, cells phosphorylate or add phosphorus to an ADP, or adenosine diphosphate, to make ATP, or adenosine triphosphate. This can be done in a few ways. In substrate-level phosphorylation, a phosphate is transferred from an organic compound to ATP. Oxidative phosphorylation is used in respiration, where electrons are moved to phosphorylate ATP. Photophosphorylation uses the energy from light to derive the phosphorylation. Enzymes are organic catalysts that increase the likelihood of a reaction without being permanently changed in the process. Because they are not changed, they can be used in future reactions. There are several classes of enzymes based on the types of reactions they are involved in. Hydrolases will catabolize molecules by hydrolysis, or adding water in the decomposition. Isomerases will rearrange atoms without adding or removing anything. Lyases, or polymerases, are used in anabolic reactions to join things together. Lyases will split large molecules but do not use water in the process. The oxidoreductases can either be catabolic or anabolic. They will remove an electron to oxidize or add an electron to reduce a substance. Transferases will transfer functional groups in anabolic reactions. Enzymes are not always in their active form. The apoenzyme is the protein portion of the enzyme that is not active unless it is bound to a non-protein cofactor. Cofactors are inorganic ions such as iron, magnesium, zinc, or copper, or organic coenzymes. Coenzymes are vitamins or contain vitamins, which are substances required for metabolism but cannot be synthesized. Therefore, they must be consumed. We don't actually derive energy from consuming vitamins and minerals, but they allow us to derive energy from other nutrients we break down. The holoenzyme is the active enzyme formed by the binding of apoenzyme with its cofactor. RNA can act as an enzyme in ribozyme. A certain amount of energy is needed, or the activation energy, to trigger a chemical reaction. The activation site is the enzyme's functional site. When the enzyme functions, it will associate with a specific substrate molecule. It will bind to form a temporary compound as the enzyme-substrate complex. The bonds within the substrate are broken to form new products, or new bonds are formed in anabolism. Then the enzyme dissociates and resumes its original configuration. Activity of enzymes can be influenced by several things. An increase in temperature will tend to increase the rate of the reaction, but if it gets too high, it may inactivate the enzyme. Each enzyme will have an optimal temperature for the quickest rate of reaction. If an enzyme denatures, the non-covalent bonds break and the protein loses its three-dimensional structure and is no longer functional. Along with too high of a temperature, extremes of pH may also denature enzymes. Here we have a video animation of enzymes working, which is also going to be available to you to watch outside of this lecture in Angel.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses encourage a link between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate.
Enzyme activity can be influenced by the concentration of the enzymes as well as the substrates. As the substrate concentration increases, enzyme activity will increase because there is greater likelihood of the enzyme active site being bound to the substrate. The rate will increase until it reaches a saturation point, allowing the enzyme concentration to be a way to control reactions. Inhibitors will block the activity of an enzyme. A competitive inhibitor will fit into the active site of the enzyme, preventing normal substrate binding. A non-competitive inhibitor will not bind to the active site, but to an allosteric site elsewhere on the molecule, which alters the shape of the active site. With feedback inhibition, products are used to control enzyme activity. The products can act as allosteric inhibitors, so the more products made, the more inhibition. This is known as metabolic feedback inhibition. Here we have another animation.